Hello, everyone. I'm going to talk about which novel delivery devices, for example, implants and rings, and novel formulations can we expect in the near future. I'm going to talk first about subcutaneous antiretrovirals. This may not seem like a novel delivery system, but in fact, we've only had one subcutaneous ARV to date, which was enfuvertide or T20, and we're going to have more in the near future. Specifically, we're going to have lenacapavir, previously known as GS6207. It is a first-in-class selective HIV-1 capsid inhibitor. It's available in both oral and subcutaneous long-acting formulations. And what you can see on this slide are the results of a phase 1 pharmacokinetic study conducted in HIV seronegative volunteers, showing that at the highest dose tested, 900 plus milligrams, given either as 3 1 milliliter subcutaneous injections or as 2 1.5 milliliter subcutaneous injections, concentrations of lenacapavir exceeded the target, which was greater than or equal to 6 times the inhibitory quotient for up to 6 months. And so this is a formulation that could be given as infrequently as once every 6 months. And I think we are going to see this formulation studied for both prevention and treatment. Specifically, at a presentation given at CROI this year, lenacapavir, when added to an optimized background regimen, was shown to significantly decrease HIV viral load in heavily treatment-experienced patients with multi-class resistance as compared to placebo. So nearly 90% of participants in this study met the virologic response criterion of having a greater than 0.5 log copies per mil decrease in their viral load as compared to the individuals randomized to receiving placebo. And I believe we will continue to see this product being used for treatment of heavily experienced people living with HIV, in addition to other possible indications. I want to turn next to novel intravenous antiretrovirals. Again, this may not seem like a novel way to deliver a drug, but in fact, we have never had intravenous antiretrovirals. Specifically, what I believe we're going to see is greater use of intravenous, broadly neutralizing monoclonal antibodies. This is a figure simply indicating that there are a lot of these products in development. In fact, in this slide from the Vaccine Research Center at NIH, the circled products are those that are already in clinical development at some phase. Now, one of the nice things about these antibodies is they can be developed as long-acting formulations. And the way that's done is with a specific 2 amino acid change in the Fc binding domain of the monoclonal antibody, indicated in this figure from an article published in 2019, showing that as compared to an unmodified monoclonal antibody, in this case, a VRC01, there's a substantial slowing of the clearance of the antibody after only a single injection, such that at week 12, the concentration of antibody is nearly, well, there's more than 10-fold higher than the unmodified form of the antibody. It's important to note, however, that a closely related antibody, VRC07LS, with the same amino acid change, did not have the same impact of decreased clearance as compared to VRC01. But regardless, both of these antibodies could, in theory, be given intravenously once every 3 to 6 months. Now, VRC01 has already been studied in large randomized prevention trials. This is a study from the HIV Prevention Trials Network showing the sites that participated in a study of VRC01 at 2 different doses for prevention as compared to placebo. So nearly 2,000 patients in this study. And in a study conducted in North America, South America, and Switzerland, nearly 2,700 patients in this study, which had the same design as the previous trial, allowing the data to be combined. And when those data were combined, disappointingly, there was no overall benefit of VRC01 for PrEP in these two studies, either analyzed individually or in a pooled analysis. And remember, these are individuals coming into the clinic once every month for an intravenous infusion. And the adherence to these trials was surprisingly excellent. However, when these same data were reanalyzed based on the likelihood of the virus to which the patients were exposed being sensitive to the antibody, and that's indicated here by the in vitro neutralization level of viruses in the regions where the study was conducted, and where the neutralization showed sensitivity of less than 1 microgram per mil, which is considered sensitive virus. Those patients in those areas actually showed a significant improvement in infection and prevention efficacy as indicated by this sub-analysis, with a roughly 70 to 80% reduction in likelihood of infection as compared to those individuals in areas where virus was resistant to VRC01. And so that's indicated in these two subgroups, 1 to 3 micrograms per mil and greater than 3 micrograms per mil, where there was really no benefit of the VRC01 intervention. And these studies were published earlier this year in the New England Journal of Medicine. So I think that provides some optimism that VRC01 and related broadly neutralizing antibodies given intravenously could be used for prevention and possibly combined with other drugs in treatment. Now I want to move to topical antiretrovirals. Again, to date, we have had no topical antiretrovirals, but we do now have one topical antiretroviral, although for prevention only, and that is the dapivirine vaginal ring. Now, I have to point out that the dapivirine ring is less effective than other prevention strategies in randomized clinical trials. And part of the problem, of course, is the device is removable, and therefore it is susceptible to non-adherence. And the efficacy of the dapivirine ring for prevention is clearly related to how well the participants adhered to leaving the ring in when they were supposed to. And removal is associated with reduced efficacy in randomized clinical trials. One of the positive things about a dapivirine vaginal ring, however, is that it can be developed as a combination technology with topical hormonal contraception. Those studies are currently in process, and there is hope that this will improve adherence to leaving the ring in when it should be left in. Finally, I will note that the implementation strategy for the dapivirine ring is quite unconventional. This is a product that has been developed strictly in a not-for-profit setting and not by a traditional for-profit pharmaceutical company. And that may lead to some interesting difficulties in making the product widely available at a reasonable price. The product is also regulated as both a drug and a device, which will complicate its regulatory landscape as well. Now I want to move from topical ARVs to implantable ARVs. And of course, there are no implantable ARVs yet either, although we certainly have experience with implantable hormonal contraception. Why would one develop an implantable long-acting antiretroviral? Well, there are potential advantages over long-acting injectables like cabotegravir and rilpivirine. For example, the long-acting implants are removable, particularly in their inert or early bioerodible forms. They provide more consistent and predictable drug release. Their pharmacokinetics are not dependent on the injection site, and they may remain in place for years. For example, the inert, non-degradable subcutaneous hormonal contraceptive implants for levonorgestrel can remain in place for up to 5 years after a single implant. However, they have some potential disadvantages over injectables. There is a specialized device required for insertion. There is a minor surgical procedure to remove them, and they should be removed if they're not bioerodible, because one cannot tell from palpating an implant if it still has drug or not. Implants are also regulated as both a drug and a device, and that may provide some difficulty in moving implant formulations into a low-cost generic marketplace. The one antiretroviral implant in development currently is the islatravir implant. It uses a technology already marketed by Merck for Nexplanon, a hormonal contraceptive. It uses exactly the same polymer. It is removable. It is a non-erodible polymer, and it uses the same application device as the Nexplanon implant. There is evidence that the pharmacokinetic profile from such an implant could release islatravir for a very long period of time. And it's important to point out that islatravir, as a very potent drug, is an appropriate agent for implant technology like this. These are clinical data presented at CROI this year by Randy Matthews from Merck, looking at 3 islatravir implants in HIV seronegative volunteers. And in this study, the implants were implanted at day 1 and then removed at week 12. And what you can see is that during the period of implant, that the implant was remaining at the subcutaneous site in the host, concentrations of islatravir were maintained far above the target for antiretroviral efficacy for the entire duration of the 12 weeks, with very little decay in concentrations, in this case of the active metabolite, the islatravir triphosphate inside peripheral blood mononuclear cells, particularly with the implants that had the higher doses, 52 and 56 milligrams. Modeling studies looking at the likelihood of what would happen to drug concentrations after 12 weeks suggest that this particular implant could release effective concentrations of drug for more than a year, with a target of 0.05 picomoles per million cells of the triphosphate. And so there has been the suggestion that this implant could be used to prevent HIV infection as a single agent for more than a year with a single implantation. Those studies are in development right now. Now, the good news about the islatravir implant is that side effects were generally mild and local. And so the most common side effects were local erythema, tenderness, itching, and induration. And those side effects occurred in the majority of subjects, 61% in this analysis presented at CROI, but they were always mild or moderate in severity, and there were no serious adverse events, nor were there any volunteers who discontinued their implant prior to week 12 due to an adverse event. The types of adverse events with the islatravir implants are consistent with those observed with implants used for hormonal contraception, giving some hope that this implant will be well tolerated and could be used in the future as an option for HIV prevention. And eventually, if a partner can be found for islatravir, we could have even implantable therapy for HIV. Okay, I want to finish by giving a few words about what happens beyond the near future. What will the distant future hold for drug delivery in treatment and prevention of HIV? Well, there are a number of different technologies in development currently that may have an impact on this disease. And that includes microneedle patches, shown here in the upper left corner, reduced injection volumes for drugs like cabotegravir and rilpivirine, new implant technologies for other antiretroviral drugs, including both non-erodible implants like the islatravir implant and bioerodible implants for other antiretrovirals that would be similar to the implant technology used for anti-hormonal drugs like leuprolide. As I mentioned, there are combination technologies in development for antiretroviral rings with hormonal contraceptives, for example, combined with dapivirine. And finally, there are long-lived gastric reservoirs that could be given orally, but release drugs slowly over a period of weeks. And these reservoirs have even been tested already in human volunteers. So I think the landscape for novel technology is quite exciting, and I think we can expect to see a number of these technologies coming forward in clinical trials in the near future. So I want to thank those who contributed to this presentation, particularly those who loaned me, in some cases, unpublished data and slides, especially Bill Spreen from ViiV, Richard Koop from the Vaccine Research Center, Mike Cohen from UNC, Jay Grobler and Randy Matthews from Merck, and Martin Ree from Gilead. I want to acknowledge my funding sources, and also for anyone interested in following the long-acting HIV device and formulation story, I recommend to you our website, the LEAP website for the long-acting extended release antiretroviral resource program, longactinghiv.org. And that's the end of my presentation. I look forward to your questions.